right. Hi, everybody. I'm Julie Passarelli. I'm the Exhibits and Collections Curator here at Carpinteria Aquarium. And it's my pleasure and honor to introduce tonight's speaker, Dr. Kevin Lafferty. Dr. Lafferty is a senior marine ecologist with the US Geological Survey at University of California, Santa Barbara. He earned his PhD in ecological parasitology from University of California, Santa Barbara, and did a postdoc with the National Marine Sanctuary. His research focuses on marine conservation in estuaries, sandy beaches, kelp forests, and coral reefs with a special fondness for parasites. Who doesn't have a special fondness for parasites, right? He studies how parasites affect ecosystems and in turn how ecosystems affect parasites. His work on human diseases such as malaria, schistosomiasis, and toxoplasmosis has relevance for how human health responds to global change. He has published over 170 peer-reviewed articles, several book chapters, and edited 2 volumes for the Transactions of the Royal Society, one on food webs and the other on marine diseases. His research has been featured on NPR's Science Friday, and he has given a TED Talk. He was American Society of Parasitologists 2009 Henry Baldwin Ward Medalist and is among the few USGS scientists listed as highly cited researchers, with his work being cited more than 2,000 times per year. The title of tonight's talk is Parasites and Food Webs. Please help me welcome Dr. Kevin Lafferty.
I see that most of you probably didn't read the title of the talk. It came anyways. Julie's been asking me to come down and give this talk for a while, and I'm so grateful to return what I think is a really big favor that I owe to Cabrillo. And I remember back, it was probably about 1975, getting on the bus from Pasadena to come down to a junior high school field trip to what was then the only aquarium, and really being enthralled and transfixed by, all this marine life that was, you never saw it on the Jacques Cousteau special on Sunday evenings, right? This was the stuff that was right out here. And that was really special for me because it made me want to go see for myself. And only a few years later, I learned to scuba dive, and I became really fascinated by all the marine life and decided that I wanted to become a marine biologist. And to a certain extent, that career change in the bulk of my life could probably be tied down to the impact that a field trip in junior high to, this aquarium had. And I think that, if you're a friend and you're a donor and you're a supporter, that's really what your hope is, that you have a chance to change young people's lives, to experience the ocean, and to have some sort of special relationship with it. So anyways, I'm very glad to be here and to talk about one of my passions, which frankly wasn't really about parasites. It was mostly about marine food webs. And I'll ask you, is anybody able to recognize this species? So wahoo or ono, if you want to use the Japanese name. This is, for me, this is like the romance of the food web. You have the bait fish that are trying to get away and this incredibly beautiful predator going after it. And in the oceans, as in all ecosystems, this is sort of emblematic of the struggle for life and death. For the food, it's really critical whether it gets away, and it's also critical for the predator that it gets to eat what it needs. And ecologists have long been fascinated by these types of consumer predator-prey interactions. In fact, really one of the underlying, I would say, themes of ecology is what we call the food web, which is one way to sort of take something like this wahoo and understand what its place in the ocean ecosystem is. It is feeding on things and it's being eaten by other things. And these food webs that ecologists have put together have been described as ecological maps. They help us trace the flow of energy through systems. They help us understand things about the fisheries that we might depend on. They help us understand what effects we might have on systems if we take species out or we put species in, etc. So think of this as sort of our map to the natural world and how important maps are to all of us. Now, I spent most of my career before finishing my PhD thinking about marine food webs and fascinated by them. And when I decided to study marine biology, I went through a bunch of different systems, and I'll mention later how I got into parasitology. I'll tell you now, actually, because it's a story that I think my friend Kimo, who's here, will appreciate. So I joined a lab that studied parasites, and I told my advisor at that time that I wasn't interested in parasites at all. I found them disgusting, as you rightly should. But at one point, I didn't have an option for a job in grad school, and the only thing I had available was to teach the parasitology course. And I'd never taken it. I was not interested in it. It was one of the most horrible experiences of my life, maybe only second to the horrible experience of my students having a TA that really didn't know what I was talking about. But one of the things that struck me was that I thought I had a master's degree in marine biology. I had a bachelor's degree in marine biology, and then I walked into this classroom, and we would bring the sea urchins and the cucumbers and the sharks and the fishes to the students, and we would cut them open, and we would look inside, and there were all these parasites. And nobody had ever mentioned that there were parasites in the ocean to me before, and I thought I knew everything. And here I was, oh wow, there's this whole mystery that really remains to be unraveled. So that's what I've spent about the last 25 years of my career working on. So about this time when I was getting into parasitology, there were a couple of prominent Canadian parasitologists, and they wrote, oh, wait a second, I'm sorry. No, I wanted to tell you what our lab does with respect to these food webs. So instead of just studying the food webs themselves, here we are deep sea fishing for that ono that's in the food web. It's my grad student, John. Okay, so that's the onos. In the ono stomach are these parasites. And as parasitologists, you know, that's gross to you, but we really think that's cool. And I was saying I have these two friends, they're Canadian parasitologists. And in a sense, I think because they're Canadian, they have this sort of chip on their shoulder, like that nobody really pays attention to them. And so they also were kind of upset about the fact that ecologists had published over 200 papers on food webs, and none of them had parasites in them. And so they're Canadians, they have this chip on their shoulder, but they're also very polite. And so they published this paper with this lovely title. It says, "Food Webs, a Plea for Parasites." And I put the flag up there. They didn't put that in. But what David Marchiesi and David Cohn wrote was, listen guys, you built these roadmaps to ecology, okay? And those roadmaps don't have parasites in them. In a way, it's like you've built roadmaps with the freeways, but no surface streets. And so how are you going to get where you really want to go, which is this understanding of natural systems, if you can't drive on the surface streets, you have no maps to use them? And they had the following points. They said, listen, firstly, there's more parasite species out there than free-living species, so let's give them some respect. Secondly, it's quite possible that parasites are affecting some predator-prey interactions that are important in food webs. And thirdly, it's probably important to think about parasites in food webs just from the fact that a lot of the parasites that we're exposed to come to us from food and their integration into food web. So anyways, when this paper came out, we were really inspired. We were like, gosh, that's right. You know, parasites aren't in the ecological literature, and we thought about how can we put them in? And we decided that we could do this because we'd been working in this small estuary up in Carpinteria called the Carpinteria Salt Marsh. And for many years, our students and I had been doing lots of different projects and dissertations and so forth. And so we really knew what the species were. And one of the great things about working in a salt marsh, which makes it easy to study as a food web, is you know when you're in the salt marsh because your boots are covered with mud, and you know when you're out of the salt marsh. So you can define the actual extent, the in and the out of the food web. And also, food webs aren't coral reefs. It's not like there's 1,000 species that you have to worry about. You can wrap your head around the diversity of a salt marsh. And also, it's not 1,000 feet underwater. It's just, at low tide you go and you can observe what you want to do. So anyways, going into Carpinteria Salt Marsh, and with our background and our students' help, we knew there were a limited number of free-living species that we were going to have to worry about. 39 really common bird species, 8 fishes, 29 vertebrate taxa, and 12 plants and phytoplankton, things like that. So once we had this list of the most common species, then to build a food web is fairly straightforward. You just figure out who eats whom, and we call these feeding links. And we can do this by direct observation. So here's a Pachygrapsis eating a mudsucker. That's for you, Ralph. Then we can also go to the literature and find out what has been reported that things eat, and we can look at gut contents and so on and so forth. So we can assemble from the species list and the feeding links the roadmap for Carpinteria Salt Marsh, and here's what that roadmap looks like. And so on the one hand, you're looking at this, you're saying, that's not a roadmap I want to use to get anywhere, right? It's really messy. But let me explain to you the layout of it. So down here at the bottom are the plants. On the top are the birds of prey, okay? And then fishes and so forth in between by what we call trophic levels. So herbivores, things that eat herbivores, et cetera, up the chain, okay? But it's not a food chain. You can see it's a lot messier than that. When ecologists study food webs, we take an incredibly messy picture like this, and then we try to deduce certain sort of statistics of it so that we can quantify its complexity, for instance, and compare it to other food webs and so on. And I'll talk a little bit about that more. But of course, we wanted to fulfill that Canadian dream, which is to put the parasites in the food webs, and so we did that again with the help of our students. And there's about 44 common parasite taxa that we find in those species from Carpinteria, and these are the different groups that they come from. And so if we take that roadmap and we add the surface streets in there, this time in red, we see the food web looks really different. And when we published this, really what we argued was, hey, ecologists, your 200 food webs, they weren't complete. You were missing a big part of the story. So, but giving talks about this, if I was trying to make the case that parasites were important to a group of scientists, then usually the refrain that I would get back was, well, okay, fine, there's a lot of parasites in there, but they're small. They're, that we could ignore them because they're insignificant. And so we thought, okay, how do we assess that? How do you assess the importance of species in an ecosystem? And we brainstormed about it, and we used what one is one of the few ecological laws. And this may sound technical, but it's something you already know. If you plot the body mass of a species against its abundance, and we do this on a logarithmic scale so that it forms sort of a straight line, what we find is that really big things are not as abundant as really small things. Okay, this is not, shouldn't be that surprising to anybody. And in Carpinteria Salt Marsh, the red squares are the birds, the blue squares are the fish, and the green pluses are the invertebrates. And you can say they span a wide range of body sizes and abundances, but basically, you can fit it to a line that has a negative slope, okay? And Ecologists care a lot about the fact that that negative slope is not exactly equal to -1. Okay, that's really not that important, I suppose. But basically, again, fewer elephants than mice. That's the ecological law from this, what we call allometric scaling law in ecology. So because we had a food web with all the free-living species, we could ask the question, where do parasites fit on this? Okay? If parasites aren't that abundant, they're going to fall off down here. In other words, they're going to be a lot less abundant than we would expect given their body size. And in fact, that's sort of what we find. The parasites are less abundant than free-living species of the same size. And so we were a little disappointed that our critics were kind of right. And so what that meant is that we thought a little bit harder about it. Now, mind you, I mentioned to you that we have really one law in ecology, and that's this straight line. And what this told us, what it suggested to us, was that law was actually wrong. In other words, it's not just the body size of organisms that determines how abundant they are. There's something else. And that should also be intuitive to you. That something else is their trophic level in the food web. So if you go out to the Serengeti, lions and zebras are about the same size, but there's a lot more zebras out there because they're eating grass, and the lions are eating zebras. So if you statistically control for trophic level and body size, then you get a different picture. What it says is that parasites and free-living species all fit on the same line. That's the real ecological law, that abundance is determined by body size and trophic level together. And really what this is saying is that those parasites seem like they're less abundant than they should be, but that's because they're at the top of the food chain. It's not the lions, it's the lions' parasites that are up there. So parasites taught us, I think, a really important aspect about the one ecological law that we have, and that is that we needed to add trophic level to it. It also suggests that there's just as much parasite abundance and biomass out there as you would expect for free-living species. And let me give you an example. If you were to go out to Carpinteria salt marsh and you were to look at the birds using a different metric than we normally think about birds. Normally you count birds, okay? But in this slide I've plotted birds in terms of kilograms per hectare. That's how you count like chickens on a poultry farm, but we'll count shorebirds that way. And basically what you can see is in the summertime, we don't have that many birds relative to winter when they come through during winter migration. We can express that in terms of kilograms per hectare. And then we can take I think this is an example that we all agree that the birds are important members of the salt marsh community. They're the important predators. And then we can compare this to a community of parasites that's also common in the salt marsh. And we can measure those in terms of kilograms per hectare. And when we do that calculation, we find that these parasites called trematodes, that I'll talk about more later, are actually a lot more abundant in terms of the kilograms per hectare than the birds in the salt marsh. And I would argue that if you could see trematode parasites with binoculars or spotting scopes the same way that you see birds, you wouldn't pay attention to the birds. That's where the action is in the salt marsh in terms of that top trophic level. Well, let me tell you a little bit more about trematodes. They're fascinating for so many different reasons, not just their biomass, but because they've evolved these incredibly complex life cycles. The most common trematode in Carpinteria salt marsh has this life cycle. It lives as an adult worm in birds, and then the eggs of the adult pass out with the bird's feces onto the mud flat, and they infect a snail that's very abundant. I'll talk about the snail a little bit more later. The trematodes grow up, they eat the gonad of the snail, and they stay in that snail for the rest of the snail's life, which might be 10 to 15, 20 years. But every warm day, they pump out these free-swimming, tadpole-looking cercariae, they're called, a larval form that goes out into the water column and searches for the next host in the life cycle, which is a fish, a killifish specifically. And then the life cycle is completed when a bird comes and eats that fish. Okay? So that's the trematode life cycle. Now Now, what's I think particularly interesting about this particular life cycle is where the parasite lives in the fish. There's a photograph of a killifish, and if you dissect the head off the fish and you look down, peel off the skull, and you look down on top of the brain. So like here's the right eye, the left eye would be down here, the nose of the fish is here, and this is the brain. There's the brain stem. And covering the surface of this fish's brain, right here, are literally 2,000 baby worms. Okay. And so there's the hint. The hint is, what do these baby worms need to do to make it on to the next phase of life? And that is, remember, they've got to get to a bird. And to a certain extent, where they are is sort of sitting in the driver's seat. And so the question we had was, are they sitting patiently or not? So at that time, in our lab, we had a lot of interest from various undergrads. One of them's here today, Chemo. So I get to tell your story, man. And Chemo's here with his family. This is going to be fun. So anyways, so Chemo and April, you're going to know exactly what I'm talking about. Like, you know, he's just got so much enthusiasm that you don't know what to do with it. What do you do? What do you do with it? Right? I mean, and so Chemo come into my office all the time. I want to do this, I want to do this, I want to do this, I want to do this. And I was like, okay, Chemo, we need to get something for you to do. So we went out and we caught a bunch of these fish in the estuary. And now this is the way I remember it. You could tell your version. And I said to Chemo, okay, here's what I want you to do. Could you just spend 2 weeks watching these fish in this tank? So there's about 60 fish in a tank. And what I want you to do before you come back to my office is try to think about things that those fish are doing that are attracting your attention. Because what I figured was, if the fish were doing something that attracted Chemo's attention, maybe they would also be attracting a bird's attention. And so Chemo came back to me, and here was his list of stuff that the fish did. They went to the surface, they shimmied, they jerked, they contorted, they scratched. These are all his definitions of what they did. And sure enough, if you go to Carpinteria salt marsh, you can sit and you can watch these killifish. And I know the video is not great, but I want you to ask, there they are, if you're a bird, which of these fish are you going to eat? Be patient. Okay, so there's your bird. That's a little snowy egret. Now, unfortunately, the video it wasn't quite dark enough. The resolution's a bit bad. But once in a while, those fish would do those things that Chemo saw. In particular, they would sort of roll over on their side. Okay. So when Chemo came back with the list, I thought, okay, what am I going to ask Chemo to do next? And we talked about it. And then that was the easy part, right? Watching the fish. That was the easy part. But then I asked Chemo to do, which, I didn't tell you at the time, but I really thought this was not going to work. And that was that what I asked Chemo to do was to find a fish in the school and follow it for a half an hour. Now, mind you, at least to me, all these fish look exactly the same. So follow that fish for a half an hour, and every time it did one of those conspicuous behaviors, to make a little check mark and keep track of it. And then catch the fish, and then dissect the fish, and then count all the parasites on the brain. And when Chemo came back with the results, I was like, okay, now we have something. Because here's Chemo's results. So this is the number of parasites that Chemo counted on the brain of the fish. And this is that number of conspicuous behaviors that fish did while Chemo was watching the fish. All right? So uninfected fish with no parasites, did some conspicuous behaviors, but the ones that were infected did 4 times as many, and the ones that were most heavily infected did the most conspicuous behaviors. we didn't expect that we would see a pattern this strong, that this fish, even though the parasite's sitting on the brain, that this fish would be so altered in its behavior. And by the way, these are fish that look completely normal in every other aspect. They're perfectly healthy in every aspect, but these behaviors, these conspicuous behaviors that they do. So I should mention that at that point in time, there were dozens and dozens of papers that had looked at similar things, that had found that hosts that have parasites have different behaviors than hosts that don't have parasites. Either because maybe they were feeling sick, or because they were injured, or maybe because the parasite was manipulating their behavior. But what there was very little, almost no evidence for, was that these manipulations in behavior actually were in the parasite's favor. And by that I mean specifically making this fish more likely to be eaten by birds. And in talking with Kimo, we figured we could probably do that experiment. And so what we did was we went out and we caught more fish, and we asked, are birds eating more infected fish than uninfected fish? And we did this by setting up 2 experimental pens. These are big, like seine nets that form this large pen, and we put up 2 of them. We put 200 fish in each pen, some infected, some uninfected, and then we covered one of the pens with a netting that kept the birds out. And then Kimo's job was to watch the pens for the next several days, and so he'd come back to the lab, and I'd say, well, did you see anything? He's like, nope, no birds. Next day, no birds. Next day, no birds. And then one day, a great blue heron stepped into the open pen and caught a fish, and then it caught another fish, and then the next days, more of its friends came and caught more fish. And so after about 10 days, we decided to pull the pens in because we figured we didn't want the birds to eat all the fish. And then Kimo went and dissected all the fish and could compare what the fish ate versus what they didn't eat. And so by doing a little bit of math to make that comparison, we could ask what types of fish the birds ate. So this is what we estimate the proportion of fish eaten by different categories. Uninfected fish, they ate hardly any of them. Okay? And they ate almost all the heavily infected fish. Now, to get this sort of pattern, an infected fish needs to be 10 to 30 times more likely to be eaten by a bird. Okay? So we had a parasite that manipulates behavior about 4-fold, increasing conspicuousness 4-fold, and increases predation rate 10 to 30 times. That's a very powerful effect on probably one of the most common links in our food web in the estuary. So to a certain extent, the parasites are determining who lives and who dies in the fish population in a way that benefits them. And they do it not by compromising the fish's health, but by doing very specific tweaks to their behavior that attract that snowy egret to come down and eat the fish that are infected. Okay. And that was your first paper, Kimo. Congratulations. There's reprints of it. They have the journal in the back. It's pretty awesome. Okay, so I want to just show you another one. This is a video taken by a friend of mine, Fred Thomas from France. And this is a cricket by the swimming pool in the south of France, which makes sense until you see the cricket jumps in the pool, which is really not a good idea for a cricket. And the reason why becomes apparent when you watch what comes out of the cricket. How did that fit in there? Okay, so that's a parasitic nematomorph. The common name is a horsehair worm. And they have an unusual life cycle also. They just use the crickets for a final host. They use another insect as an intermediate host. I won't go into that detail. But when they get into the cricket, once they're mature, that parasite wants to go to a pond or a stream so it can find other worms to mate with. But crickets don't go into ponds and streams. So the parasite is able to alter the neurotransmitters in the cricket in such a way that makes it super thirsty, and it jumps into the water, at which point the worm explodes out of the abdomen. Okay. So that's just creepy sci-fi stuff, but we're interested in, does that mean anything at an ecological level? It's cool from an evolutionary perspective, but what does it mean, for instance, to streams like this in Honshu, Japan? And so I started working with these Japanese trout biologists. And they were working on an endangered species of trout called the Kirakuchi char that had been overfished and its habitat had been wiped out. And it was so precious that they have all the fish tagged. Every month they go visit them. They capture them by electroshocking. They put a tube down their throat and pump their stomachs. They figure out which ones are gravid. They can calculate growth rate, et cetera, et cetera. And they knew more about the individual char than we know probably about most other species that are studied. And what was cool was that, like most trout, they knew that the trout swam around and eat bugs at the bottom of the stream. But in September and October, that all changed. Instead of eating the bugs on the bottom of the stream, they were feasting on crickets, and their stomachs were full of crickets. So the idea was that here we have this parasitic worm. It's in crickets. The crickets are jumping into the stream, so the worm can get into the stream. But that's great food for a trout to have some cricket flailing along on the top of the surface of the stream, and so they totally changed their diet during that period. Now, it turns out that the worm has evolved to deal with this. It almost always gets out of the fish. When the fish eats the cricket, it gets out through the gills or through the mouth or even through the anus. They're really tough worms, and they get out. Okay, so again, that's pretty interesting that the fish are eating the crickets and so on. But because they had been doing so much careful documentation, they could actually figure out how important this was to the trout's diet. And when they do the calorie estimate by different prey type in this food web, what they find is that 18% of the calories that this fish gets are from insects that are on the bottom, 22% from insects that fall into the stream, and 60% of the calories that this endangered species gets for the whole year are delivered to it in the form of crickets committing suicide by jumping into the water. So that, I think, is, for a single number, that should be the most impressive, evidence that I can show you that parasites do have strong effects on food webs. And then furthermore, that has effects on what the food web looks like. So the simple food web in that stream, you got fish feeding on insects, and the insects are eating the leaves that fall on the bottom, and that affects things like nitrogen and algal growth and so on and so forth. And when you throw in the cricket into the system and the parasite, that changes a lot of the actual details and the dynamics of the way that food web works. So the food web is completely altered by having the parasite in the system, much to the benefit of the endangered species, to the point where they make the argument in the papers we've written that without the parasite, this fish wouldn't be able to persist. So if we go back to my Canadian friends and their plea for parasites, I think that they got the most interesting question, which is what happens when we put parasites into food webs. But I'm interested in the reverse question as well, which is, if we're going to understand parasites, do we need to know something about food webs? And so I'd like to talk next about parasites, a plea for food webs. And trust me, people that study parasites are 99.9% interested in just killing them and getting rid of them, and not so much thinking about their broader role and how food webs might affect them. And so I'd like to go to our local system, kelp forests off of California. And so some of you probably recognize the purple urchin, Strongylus intratus purpuratus. And my colleagues that work out in the Channel Islands, Santa Barbara Channel Islands, have been monitoring all sorts of aspects of the kelp forest community for many, many years, and we have one of the best data sets to understand dynamics and food webs of that system. But first I have to show you something that's not really about food webs so much, but it's the relationship between host density and infectious disease. And basically, this makes sense again, I think, that most infectious diseases are passed from one host to another through contact. And if you have a lot of hosts crowded together, then you're much more likely to transmit diseases from one host to the other. And so if we plot here the probability of a bacterial epidemic in the sea urchin population versus the number of sea urchins per meter squared, you can see that where you have a lot of urchins, you're much more likely to have these bacterial epidemics. Okay. So how does this tie into food webs? Well, in this system, we also have food web effects that are pretty well documented. So in the analyses that I've done and other people have done, we know that spiny lobsters are important predators of sea urchins, and sea urchins are important predators of kelp. And where you have a lot of lobsters, you have few sea urchins and a lot more kelp. But that's not the way most Southern California food webs look, because we are heavily involved in this food web, especially in terms of taking spiny lobsters out of it. And when we take a lot of spiny lobsters out of the system, we get more purple urchins, we get less kelp. And that's what's called a trophic cascade. And we see it happens throughout the Santa Barbara Channel Islands, and it definitely happens here in Southern California. So what we've seen is that a food web can change the abundance of an important host in the system, and that leads to direct consequences for infectious disease dynamics. And so I'm going to walk you through the data that we have by comparing marine protected areas, of which at the time we did this study there was only a few, versus the rest of Southern California, which is heavily fished. So the way that the data work is that the left-hand column are areas where species are protected from fishing, and the right-hand column is everywhere else. And so you can see up at the top panel are lobsters, very few lobsters, except in the marine reserves where there's lots. In the marine reserves, few sea urchins, lots of sea urchins outside. Inside the marine reserves, lots of kelp, less kelp outside of the marine reserves. And this is really the punchline for me. Where you have fishing for lobsters, you have a lot of epidemics in the sea urchin population. So it's an indirect effect of fishing promoting infectious disease transmission in this particular species, because where you have a lot of urchins, it's easier for these diseases to spread. So this sounds like I'm saying that fishing causes disease, and we wanted to go and see how general that was. So we went to the Galapagos, because you need an excuse to go to the Galapagos. And here we have a really similar type of food web, or at least a simplified version. So we have fishers, we have large predators, we have sea urchins, and we have algae. And work that a student I'd been working with showed also that we have these trophic cascades too. In other words, in reserves we have more predators, fewer urchins, and more algae. And this time we weren't not looking at bacterial epidemics. We were looking at two species of parasitic snails. They're called eulimid snails. They are highly adapted parasites to echinoderms. This one, This one infects the tip of the spine, and it forms a gall, and it feeds on the epithelial tissue. This one sticks its proboscis in through the test of the urchin and sucks the gonad out. Okay? And so, for instance, if you look at the reproductive gonad index for urchins and you plot it against the number of snails it has, there's a pretty strong effect of these snails on the reproductive output of the urchins. They have a very important effect on the sea urchins, perhaps even stronger than those bacterial epidemics that I'd been measuring in Santa Barbara. So then the question is should we have the same result? We have fished areas in the Galapagos, we have unfished. Should we get more of these parasites in the fished areas? And that's what we predicted, but when we look closer at the food web, there was one additional species that we found makes a big difference. And these are these little spider crabs, and the spider crabs love to hang out in those pencil urchin spines. And their favorite food are the snails. So they are on these, and you don't usually think of predators eating parasites, but that's exactly what's happening. If you look at the number of snails per urchin, with and without crabs, this is without crabs, more than one snail per urchin, and with crabs, you hardly ever see snails on that sea urchin. Okay? So we throw this additional species into the food web, just one small change, and then when we look at the interactions among all these species in the food web, we call these red arrows are negative interactions, the black arrows are positive interactions, and the details aren't that important. It's just to show you that this one species really messes stuff up for us in terms of changing our expectations. And so overall, what we find is that the relationship between fishing and snails is a red arrow. It's negative. We have a lot less parasitism with fishing than we have without fishing. So we have the exact opposite relationship between fishing and parasitism, just due to the addition of one species to that food web. So those details matter a lot. Okay, Galapagos. Let's go from the Galapagos to just about the furthest place you could go, which is the Arctic Circle, Takvatn in Norway near Tromsø. And I've been working with these Norwegian limnologists, and they've been really interested in taking our approach and putting parasites into the food webs that they study in these Arctic lakes. And when we've done that, we get a similar sort of picture to what I showed you in Carpinteria salt marsh. Lots of red lines showing parasites coming in and interacting with the fishes and the invertebrates and the birds. What was interesting to me about Takvatn was that they did what you might call a biodiversity augmentation to this lake back in the 30s. Takvatn was a sort of place, if you know Norwegians or Swedes, you know they're super into cabin culture, and they go and they have their fishing cabins and they do ice fishing and all sorts of snow things. And so Takvatn was a famous lake for fishing brown trout, and there were all these cabins around the lake. People go fish brown trout. And then in about 1910 or 1920, gill nets were invented, and they started putting the gill nets out for brown trout. And then after a while, there weren't many brown trout anymore, and these people went up to their cabins. They didn't have anything to fish, so they thought, all right, well, let's throw in Arctic char. So they went to the lake next door, which had Arctic char that their lake didn't have, and they were probably been jealous about it for a long time. And they brought the Arctic char over, and they put it into their lake. And the Arctic char didn't do very well, maybe because of the gill nets, but they assumed it was because the Arctic char didn't have anything to eat. So then they brought stickleback in and added those to the lake. So we take a system that's relatively simple, and then we just add 2 new species to the food web. We make it a little bit more diverse. And then the question is, what happens to the parasites? And by sort of doing retrospective ecology, what we find is that we got a lot more red lines in here. Because we've added these 2 fish species, there are 5 parasites that can now complete their life cycle in that lake, in addition to 5 different bird species that can now come in and feed on stickleback that weren't able to feed there before. So this is an example of how parasites really depend on biodiversity to complete their life cycles. And it creates this really counterintuitive prediction that healthy ecosystems are ones that are rich in parasites. If you think about it, it makes sense, right? I mean, the parasites need to move around the system, and the more options they have, the better they're likely to do. But it's not a really popular hypothesis, I have to say. And so we decided to go back to Carpinteria salt marsh and ask, can we see associations between biodiversity of free-living species and biodiversity of parasites? Now in Carpinteria, the thing that we like the most in terms of biodiversity is this incredibly rich community of birds. And of course, if you remember that life cycle diagram I showed you earlier, you recall that those birds are the final host for the adult trematodes that go through that fish, and lots of other parasites in the salt marsh. So I showed you the life cycle with killifish, but there are 20 different species of trematodes in Carpinteria salt marsh, and each one has a slightly different life cycle and ends up in a different type of bird or set of birds in the salt marsh. So the prediction is, if you have an area that has many different species of birds, that creates opportunities for many different parasites, and we should be able to see all those parasites in this single snail that they all go through. So it's actually a pretty simple study to do. We just go collect the snails from the mud and dissect them and count the number of parasite species in them and ask whether that corresponds to the number of bird species that we see in those particular habitats. So here's a photograph of one of the study sites that we had, and maybe a little bit hard to see from this slide, but there's a bunch of little bumps in here, and those are the hundreds of snails that are in this tidal channel, so it's a really easy system for us to work with. And again, the idea is to quantify the bird community at this site, and then estimate the parasite community and the snails that are living at that site as well. The problem is that almost every time you go out to Carpinteria Salt Marsh to count the birds, this is what you see. No birds. And we thought, well, maybe what we need to do is put cameras out that could do a better job of that. And so we resorted to this time-lapse videography. Takes 5 seconds of video every minute for 12 hours a day for a full tidal cycle. It generates a lot of these video clips that we can then give to our undergraduates that are incredibly skilled at watching video clips. And the cameras work during the high tide when we might not normally go out there, and one of the best things about them is that, you know, the cameras are not disturbing the birds the way that we do. So that we get really good estimates of the bird community, and then when we plot the diversity of birds on the x-axis here versus the diversity of parasites on the y-axis, we get a really strong fit. The more bird species, the more parasite species. And here's what's really mind-blowing to me about this particular graph. The amount of effort that it took for us to estimate the bird species richness at one of those sites was 10 times higher than the amount of effort it took to estimate the parasite diversity at one of those sites. In fact, it just means that if we wanted to know the bird species richness at those sites, it would have been much more cost effective for us to grab the snails, dissect them, and count the number of species, and then say, oh, there's probably going to be 7 species of birds that use that site. So, but that's an observational study, and rarely in ecology do you get a chance to do sort of a large-scale field experiment, but in Carpinteria Salt Marsh we did have that chance. And what happened was that Carpinteria Salt Marsh has a nature reserve portion. It's got a big fence around it. Nobody gets in but the researchers. And then next door was an area that was open. It was degraded. It was filled in. It was full of trash and weeds and so on and so forth. And it wasn't functioning very well. So we thought, the first question was, well, do we get more parasites in the nature reserve than we do in basically the vacant lot next door? And this was the difference. So the gray bar is the parasite richness in the nature reserve, and the open bar is the significantly lower parasite richness in the degraded area. So that suggests that, you know, there's some value of that protection that allows parasite life cycles. The reason we were interested is that we'd learned that the city was going to spend $2 million restoring that vacant area, and then we could track what happened to the parasites over time afterwards. And the first thing that happened was they brought in a bunch of heavy equipment, and the parasite diversity didn't go up, it actually went down due to all that disturbance. And so we should have stopped then, perhaps, but we kept following it. So here's the heavy equipment effect, and then over time, over 6 years, the parasite diversity climbed and climbed and climbed until at the very end of our study it was actually significantly higher than the parasite diversity in the nature reserve. And so this was consistent with this idea that, you know, the parasite diversity was following the bird diversity. We went and we congratulated the city on, you know, increasing parasite diversity with their $2 million restoration effort. And I wish they'd laughed like you guys did. They didn't laugh at all. And then we explained to them, listen, the only way that you're going to get those parasites there is if your restoration was successful at bringing in the invertebrates and the fishes that attracted all those birds to come in and complete the food web elements in your new restored nature park. And that's what the parasites needed. So that was an example of, in this case, you know, host diversity being followed by parasite diversity and being able to use parasites as indicators of free living species. Okay, so that's sort of a happy story. Restoration, we get more parasites, yay. But you know, what we're in marine systems, we're more and more faced with this sort of change, not restoration, but really these, you know, serial depletion of fisheries and the change from what I call my grandfather's ocean to my grandchildren's ocean. And we wanted to ask, well, what are the effects of large-scale fishing on parasite diversity in the oceans? And we were mostly interested in the habitat that we felt would be most sensitive to that, and that's coral reefs. So we work in the Central Pacific in a place, in this map you can see, here's, let's see, Hawaii, the Northern Line Islands, etc. Boy, those maps are really hard for you probably to figure out. But basically, if you go to Hawaii and you go 1,000 miles south to the equator, that's where we work. And it turns out it's very easy to find coral reefs that are heavily fished. And so for this study, I'll show you data that comes from Christmas, which is one of the Northern Islands. It's a very large atoll. It's densely populated. And the people there are very, very poor, and they depend on food from the ocean. And so they fish it a lot. And when you do fish surveys there, you find that they have overfished the reefs in ways that are pretty degraded. It's a lot harder to find places that are not heavily fished. But to do that, we go to a place called Palmyra Atoll. And this map gives you a better idea. So there's Hawaii, 1,000 miles south. There's Christmas, there's Palmyra. Very similar fish faunas, etc. But Palmyra has never had an indigenous human population. It had some airmen on it briefly during World War II, but since then it's been pretty protected and no fishing is allowed. It's operated now as a research station by the Nature Conservancy, and that's where we do our work. So when you fly into Palmyra Atoll, we charter a plane and you land on an airstrip out there. And the first thing you do when you land is you want to get in that beautiful tropical water, and so you go snorkeling and you see the pretty tropical fish, and you look at it and something strikes you as there's something wrong here because those fish look really nervous. And it's because they're looking over your shoulder at lots of predators. Because nobody's fishing in Palmyra Atoll, it's a very intact coral reef fish community, and so the idea was that maybe that changes the food web and that changes the opportunities for parasites. So we chose 5 different species of fish: a chromis, a surgeon, a snapper, a damsel, and my favorite, the arceye hawkfish. And first we thought we would go out and catch fish the way that the aquarium collectors catch them, with a little net. And either we were just really bad at doing it, or the fish were too nervous and we had almost zero success trying to net fish. And so instead we switched over to using what we call microspears, which is a jargonistic term for a straightened out coat hanger on a rubber band. And it doesn't sound very sporting, but just try shooting like a little tiny fish with a coat hanger. It actually is harder than you think. Plus, in Palmyra, this is our study site. It just goes on and on and on like this, and the sharks are actually, they're curious, and they're not really a problem when you're shooting little fish, but when you shoot big fish, then it can get kind of scary, and we set off shark feeding frenzies and so forth, and so for the big fish, we had to stop shooting them underwater to collect them, instead go with hook and line, and even then, a good fraction of the time, the sharks got to them before we did. So because we were in a place where nobody had ever looked at parasites before, we found a lot of new things that nobody had ever seen, and there's just an example of some of the parasite diversity from the different species. And just to compare one measure of parasites from these islands, if we look at the number of species in a sample of fishes, Palmyra, which is the pristine area that's not fished, just consistently from fish to fish, more parasite diversity than on Christmas, which is heavily overfished. And we've since, my colleagues and I have repeated this in many different islands around the Central Pacific, and we get the really consistent pattern that the more you fish, the fewer parasites you get. And what this suggests is that pristine biodiversity and parasites go together, and that leads to a quandary. What do we do when those parasites are parasites that we care about personally, when they affect human health, for instance? Do we have to make a choice between biodiversity and health? And I think the answer is that we might sometimes. But the other answer is, I think if we think carefully about food webs, then we can actually find some win-win situations where we can improve food webs or restore them and reduce parasites. And that's what I want to end the talk with, is some of our work on human schistosomiasis. Now, schistosomiasis is caused by another one of these trematode worms. It has a life cycle that's similar to the ones that I showed you before. The larval stage lives in these freshwater aquatic snails, and then the life cycle goes into humans. It penetrates through the skin, goes into the circulatory system, lives in the bloodstream, in the veins, and eats red blood cells. It's actually remarkably a romantic parasite. The male worm is big, and it forms a permanent copulatory embrace with the female, and every day she pumps out eggs, and they might live for 10 to 15 years. And although that's romantic, the problem is that if you're releasing eggs into somebody's bloodstream, they have a hard time getting out. So the schistosome eggs have a little hook. They bury in through the blood vessels, or they get stuck into the liver, and over years and years and years, they accumulate in either the liver or the bladder or other parts of the body, and that leads to chronic health problems and sometimes death. So schistosomiasis might not be something you're familiar with because it's a problem in tropical countries, and therefore it's one of these what we call neglected tropical diseases. And we've been studying schistosomiasis in the Senegal River Basin. What was really interesting to us was that schistosomiasis didn't used to be a problem in this part of Africa. It was a problem down in southern Africa, but not up here in the more northern area close to the Sahel. But in 1986, there was an outbreak of schistosomiasis that became the largest outbreak ever recorded along the Senegal River, and it has persisted until this day. So one of the questions is why? And basically what we think is the problem is a thing called the Diama Dam. The World Bank funded a multi-million-dollar project to build this dam. It's not a tall dam, it's not for hydropower, it's simply to keep the tide from going up the Senegal River so that you have fresh water that you can use to irrigate sugarcane fields along the bank of the Senegal River that are owned by a few wealthy landowners. And when you built the dam, two things happened. One was that you brought in a lot of people from the south to come work in those sugarcane fields because you created economic opportunities, and those people had schistosome worms with them, and they brought them to northern Senegal. The other thing that happened was that you changed the habitat, the food web, the types of plants, and that, for whatever reason, made things really good for freshwater snails. So we went to Senegal, and we were curious about what might have happened to the food web when they put the dam in, and one of the things that we were curious about was crustaceans, in particular giant freshwater prawns called river prawns or macrobrachium. Now, these types of crustaceans, you remember I told you what the favorite food of that little spider crab was. Well, that's also true for these river prawns. They love to eat freshwater snails. And they have an interesting life history where the females go and release their eggs into the estuary, and then the juveniles have to sort of migrate back upstream, and they can do it for hundreds of kilometers, where they then become adults that feed on snails. And when you build the dam, there's no way for the prawns to get back upstream. So our thought was, gosh, maybe the dam has interrupted this food web, and that has increased snail populations, increased schistosomiasis, and that gives us a key that we might be able to restore the food web in a specific way that could benefit human health. So we did some really simple aquarium studies. And basically we can raise populations of snails in big aquaria, and this is what their population growth rates look like without prawns, and when you throw prawns in, the prawns eat most of the snails. Okay? And even more important than that, if you look at infected snails, it turns out that the prawns really love to eat them. They like to eat the infected snails more than uninfected snails. So that seems great. I mean, you have a predator, a native predator, whose favorite thing to eat is infected snails that make people sick. So the aquarium studies were enough to stimulate us to try to do a major field experiment where we tried to intervene with a human health outcome. And it was a pretty straightforward experiment on some levels. The idea was we chose 2 villages, similar villages along the Senegal River. And the Senegal is just an absolutely wonderful place for so many different reasons. But if you live in northern Senegal and you live along the Senegal River, you spend a lot of time in the river. That's where you do your laundry, you do your dishes. If you're a kid, that's where you spend your time bathing because it's 100 degrees and so on. So these people are very tied and very connected to the river through usually just a couple of sites near their village. And we thought, okay, maybe we can go in and put prawns at one of the village, what we call transmission sites, the places where they're getting their water, and we'll use the other village as a control. Now, all of a sudden, we've got ethical problems, right? We're giving one village one thing and not the other. So, but the plan was this. So we went into both villages, and we took all the kids, and we gave them drugs that cured them of the worm. So every kid in every village is treated for schistosomiasis. And then we put prawns at one of the villages and not the other village. And then we came back 4 months later, and we looked to see the rate of reinfection in those kids at the 2 villages. And then we treated them both again, too. So that was the idea, at least, being that both villages were better off being involved in our study than had we not done it. And we didn't know what the outcome was going to be. So here is a measure of infection in the kids before we put the prawns in. The purple indicates one village, and the gray indicates another village. And this is a measure of the number of eggs per gram feces in a fecal sample. It's just a measure of infection. And this is before, again, that we put the prawns in the system. And now we're going to add prawns to the village that had the biggest problem. And again, that was an intentional choice based on the ethical issues of who might benefit more. And then we measured the snail abundance over time. And what we found was that in that site where we added the prawns, most of the snails were gone compared to the site where the prawns were not added. And again, in particular, it was the snails that were infected with a parasite that really got hammered, perhaps because the infected snails were easier prey for the prawns to eat. But more important was going back to the kids and their infection levels, and that's where we saw what we'd hoped we'd see, which is in that village that had lots of infection after the prawns. Their infection rates went down during the rainy season, whereas in the control village, their infection rates went up as expected during the rainy season. And then again, the kids got treated again. So that's some indication that these prawns are an important part of the food web, and taking them out messed with that food web and created negative health outcomes for people that lived along the river. And so what we call this potential win-win between biodiversity or native restoration of habitats and human health. And we actually call it a win-win-win because a little bit of butter and garlic, those prawns are really delicious, which is important because if you live in a village in Senegal, what you want is some sort of fishery that you can sell and make some income off of. So it's not just about restoring, but it's about restoring fisheries. Now, speaking of butter and garlic, I just want to end on where we left.
Black. Thing is so good. So how many pieces we have? 1, 2, 3, 4, 5, 6. We have any Look at that sucker right there. See the sucker on that one. It's not bad.
asked my student, what's the expression on his face? Well, thank you very much.
Now that was an incredible presentation. Thank you so much, Dr. Lafferty.
Oh, my pleasure.
I learned a couple things. One thing, oh, this is for you before I forget. I'll go home with it if I'm not careful. I learned a couple things. First, I did not know snails were parasites in and of themselves.
Yeah.
I did not know that.
Ironic, isn't it?
It is. I studied snails. I know Dr. Suzanne Miller studied snails. Alan Miller studied snails. But the biggest thing I learned, I'm thankful you were not my major professor.
Yeah,
Kimo, I've got
exactly.
new respect for you. Watching killifish for how many hours? Oh, I used to watch snails crawl. I thought that was terrible. So we have a few moments for some questions from the audience.
What about the prawns that ate the snails, since they were eating the ones that were infected? Didn't they get infected also?
Yeah, it's a great question. The trematodes die if a prawn eats them. There's no way they can go to the prawn, so it's not a risk to eat prawns that have eaten a snail. Yeah, that's a key part of the deal. Yeah. This is why you want to, study your life cycles, I guess, is the point, because you wouldn't want to be, hey, local people, have some prawns that just ate some infected snails and then they all get sick. That wouldn't work. So yeah, it's not a problem.
So no, does that village now have prawns as a fishery?
So right now we have a couple things going on in Senegal. We're this is a Senegalese river prawn that's never been farmed, and we've just now gotten the life cycle cracked in hatcheries in Senegal, and so we're to the point where we can start giving little prawns to villagers, and they can grow them out. And I've also been out there designing the world's first prawn ladder to get prawns back up over the dam and hopefully be able to restore the river there. I know it sounds kind of silly, but I think it'll work. Yeah, good question.
You in the back.
In that last scene, was he eating parasites?
Yes.
What was the purpose of that?
Yeah, well, what was the purpose of that? I don't know. I mean, there was no purpose to that besides entertainment on my part and the enjoyment of you, the audience. Yeah. Yes.
So your wetland study of Carpinteria, your diversity of parasite goes up, indicating diversity of birds goes up. What's the diversity of the parasites in the birds?
Yeah, that's the hard one for us to quantify. It's really hard to get permission and ability to, and desire to shoot birds and look inside them. We do have some limited information about that, and my guess is, although I can't tell you for sure, that they probably get more parasites at sites that have more diverse prey items. But that's just a hypothesis. We haven't been able to test that. Yeah. Good question. Yeah.
So they create this additional diversity in wetlands. So if we had a parasite that was extirpated in, say, one of our Southern California wetlands?
Like, do you have one in mind?
Yeah. Do you think that we could convince a resource manager to be a really good thing to reintroduce those parasites into a wetland?
No, I don't think so at all. I mean, I think that this whole issue about parasite biodiversity and whether we should care about it, I think is fascinating. And what some people would say is there's this issue about taxonomic chauvinism. In other words, we make sort of lip service about biodiversity, but it's really only certain parts of biodiversity that we care about. And you know what? Fair enough. This is an aesthetic issue, and what people want to save is really what they're interested in. But parasites are, at least at this point, unlikely to make the cut. And I'll give you what I think is one of the most interesting examples for why that's true. When the Fish and Wildlife Service brought in the last free-ranging condors, the veterinarians that were involved in the study knew that those condors had a specific louse, the condor louse, that had flown over Southern California when there were saber-toothed tigers with the condor. And the louse is pretty harmless to the condor, and yet they did an insecticide treatment that drove that particular species to permanent extinction, intentionally. So, we've been through the question about should we, protect parasites or not, and the answer has been if we get a chance to drive them extinct, let's do it, even when they don't necessarily pose a risk to us or their hosts. So I don't think that at least in the past, we haven't seen much sympathy for parasites, and I'm not telling people how they should think about them, but as you know, many of them are fabulous examples of evolution, and also sometimes incredibly beautiful in terms of the things that they do to make a living on this planet. So I'm obviously a little sympathetic to their plight, but on the other hand, I think that as people that live in a privileged country where we're really not that impacted by tropical diseases, I can see that it's easy for us to say, let's conserve parasites, because they're not our problem. There are a lot of places in the world where they are quite a problem. In those cases, I have less sympathy for the parasites because, if we're going to choose between us and them, I kind of know where most people would put their children, on that level. So I don't know. I think it's an interesting thought experiment to think about what do you mean when you say you want to preserve biodiversity, especially when half or more of the species on the planet are parasites. Yeah.
Were there any movements or any attempts to sort of reverse the building upside down to restore the broken ecosystem?
I think the answer is no. And I think that we've seen in many, many cases that when dams go up, some people win and some people lose. And the people that win are usually pretty predictable. They're usually the people that owned the land or had the power or could make the investments. And those people are the ones in control of making decisions. So I don't think that we're going to see the dam taken out. We have started to see at least a willingness to install the prawn ladder. So we actually have permission now to do that from the dam agency, mostly because we could show it wasn't going to mess with their dam. But we've got to find a way to fund it. That's the trick. So no, I think that, and we could go on and on about dams and the ecological impacts they have and the benefits to humanity that they have also. And it's sort of, there's a cost-benefit analysis mostly that has left dams remaining around the world. Yeah. Yeah.
Extending these webs into parasites reveals tremendous complexity. Do you have any thoughts about complexity and how, just changing a couple of things have a very large number of influences and what you do with that?
Yeah, this is the stuff that keeps me up at night. Yeah, that's for me as an academic, that's the interesting question is how, to get a handle on complexity, to put a number or two to it, to understand what happens when you tweak, an element on one end of the room, what happens on the other end of the room. Can you predict those things? Are they unpredictable? And again, I agree that the parasites reveal that additional complexity, which on the one hand can be a little bit overwhelming to wrap your head around. But on the other hand, my firm belief is that we are actually capable of understanding that the details matter and we can understand them, that it's not a lost cause. Complexity is not, it's not something, it's not a complete barrier to us that it seems initially. But yeah, I'm glad that thought entered your mind because it is really what motivates me to do these sorts of studies. Yeah.
I have a question. So you said with the snails in the marsh, the worm goes into the snail and then it leaves after it dies, so the worm can be there for 10 to 15 years. So when you're doing relationships in the bird species and the worm species, could there possibly be a lag where you're seeing a parasite where the bird may coming in there for 10 years?
Yeah, that's right. So you can sort of think of the snail. So the worm is in the snail the whole time. It doesn't die. It just keeps pumping out these larvae, right? And some of those trematodes might be 10 or 15 years old. And so the question is, when did that snail get infected? Those are the birds that you're not measuring in your video cameras, right? And that's absolutely right. So you can sort of think of those snails as mini data recorders or data loggers that have been recording for the whole life of that snail. So one of the ways that we get around that is we can dissect snails of different sizes, which sort of corresponds to snails of different ages. And so we can say, yeah, it's the young snails kind of tell us what might have happened in the last 2 years. The really big ones, that's a broader integration of time. And I think that both are interesting types of data to look at. What we do see, though, is that if you go to Carpinteria Salt Marsh, I can tell you, where the spot is where there's lots of birds, and it's probably going to be the same place 15 years from now. So that those bird patterns are probably fairly repeatable in space. Time, yeah.
We have time for one more question. And then we will then convene to the gift shop and you can continue the conversation over there. So one more.
You choose it.
No, you choose it.
Oh, man. All right. I know you're going to ask it no matter what, so. Yes, I'd be happy to tell you which parasites you can get from eating fish. But let me just get this one last question, too, because you were very patient.
Well, it's just really quick, because I am really interested in the impact that we make on some of the decisions and changes that we do to our systems. And thank you very much for the topic. I think it's very fascinating. But is there a protocol that academics or researchers go through, and how far do they investigate some of these food webs to the point that they're going to know pretty much to the nth degree what kind of impact it's going to have when maybe another species is introduced or something is put in that animal's way to survive, in chances of reducing the impact of humans. I mean, is there a protocol, a scientific protocol out there that researchers follow, or is this just up to the researchers?
Oh, yeah. No, I mean, the field that I work in is very individually driven. There's no general approach to addressing these questions. So it only happens if, like, I want to go out and do it, yeah. At least so far. I mean, in other words, we don't have a systematic approach to making these sorts of discoveries that you might have in drug discovery or other types of human health issues where there's a lot more focus, I think, and funding, clearly. Yeah, good question.
Thank Dr. Lafferty.
Thank you.